Thanks, Alex. Thanks for the organizers to giving me a chance to introduce my one of my postdoc projects. This is about the link RNA in 3D genome. So we identified topological anchor point RNAs, and I'm going to be talking about it. And this is a published work. You can find it in the Genome Biology early this year. And you also can see the poster at A23. I think the introduction is not needed for this audience, but so I'm just quickly remind you what I'm talking about. That after Human Genome Project, there is an ENCODE project, of course, and then one of the striking finding was that most of the genomes are actually not really making any protein. So at that time, many people think this is just junk DNA or even dark matters, but after kind of experimental kind of techniques are really developed, and now most of people doing the sequencing, and eventually we now know that 99% are really not silent. So what are they doing? And one of the most recent paper showing that kind of non-coding RNAs are the major output of the human genome. And particularly the link RNA, long non-coding RNA, actually outnumbered the protein coding genes. I think the number is also important, but really important thing is the function. If they don't do any function, then does not mean anything. So in one of the another paper, review paper, nicely summarized what are the link RNAs are doing in our cell. So they are actually doing many things in many stages and many parts of our nucleus. So maybe you can have a look. I think most of you already seen it, so I will skip. So I'm directly going into the one what we did. Actually, this project started from some strange angle. So we actually working on some other things, and by chance we often see some kind of patterns in not only in human but also in the other species. That's quite interesting because one of the definition of link RNA is actually it shouldn't be. It's not have any sequential conservation. That means they may not really conserved throughout the species. But what we often see is that even though we don't see any sequential conservation, but we do see the kind of positional conservation. So what is positional conservation is, for example, this is SOX2 gene, this is coding gene here, very small one, and this is the longer ones are the SOX2 OT. OT is the overlapping transcript, which is a link RNA. So this is in human, but in mouse, again, SOX2 is here, and then SOX2 OT is overlapping this coding gene, and in zebrafish and others. So this is just one example. So we did see a lot of this kind of stuff. So we're just wondering whether this is just general phenomenon or this is just by chance. First question is this one, and the secondly, if this is true, then do they have any function? So we set up kind of bioinformatics pipeline to systematically identify the positional conserved RNA in human and mouse. So we called this as a pcRNA. So there are two names, pcRNA and topRNA. So again, this is published, and today I have limited time, so I can't talking everything in the paper. So I would like to focus on in the second part, which also I mostly contribute. So for the pcRNA bit, maybe I ask you to read the paper, and I would like to just summarize in one slide. So pcRNA are developmental related, which means all the pcRNA have its-associated coding genes like SOX2 OT pcRNA have its-associated coding SOX2. So in this case, most of the coding genes are really developmental related, and they do have expression in. Their expression is quite tissue specific. And lastly, the pcRNA and its associated coding genes tend to co-expressed and co-induced. So there are some other more interesting stuff. But when I did this ENCODE chipset analysis on the promoter of pcRNA and its associated coding, we kind of opened another chapter for this paper. So this is the thing where it comes from. So this is a heat map of the binding patterns of ChIP-seq. So pcRNA is here, and its associated coding is in blue. So each columns are the pcRNA, and here is in same order. So pcRNA number 1 is in first column, and in blue, pcRNA number 1's associated coding in first column, something like that. And each rows are the 2,216 ENCODE ChIP-seq samples. So that's here. So the thing actually quite striking is that here we do see kind of band, which means kind of a group of TF binding to pcRNA, most of pcRNA and its associated coding genes. So we wanted to know what is this, and of course we checked, and this is CTCF. So we checked whether this is true. So we're using the control with kind of similar length of general link RNA, and we do see definite enrichment here. Even we compared to the codings, it still have quite good enrichment. So this rang a bell that maybe yesterday there was a special session in the ISMB as well about 3D genome. So CTCF is kind of hallmark for the topological anchor point. So we wanted to check whether this is also true. So we used the high-C data. So what is high-C data about? Is that if you think the DNA in the linear fashion, this length is more or less 3 meters. But actually in the real life, this is not in this kind of shape. So this is in the very small, very tiny space in our nucleus. So that should have this kind of structure, very complex 3D-dimensional structure. So the kind of high C is from the 3C chromatin conformation capture. So 3C, and then it developed to 4C, 5C, and eventually genome-wide high C. So even these days, there are high C version 2 or capture high C, all the nice techniques. But at the time, we're just using high C. But anyway, whatever. Just one thing you should remember is that this heat maps shows clearly where is the TAD, but the thing is that the TAD location is important, but the more important thing is that this boundary is very important, because this boundary means this is really where the two strands of DNA actually bind together, and this is where many people believe there are something happening, very important thing, which is kind of gene regulatory mechanisms. So this is the very important point. And another thing is that this is very nice schematic diagram to show our chromatin having this looping structure. And again, the CTCF is a kind of hallmark to actually hold this kind of two strands of DNA, the chromatin, and actually it forms many kind of size of loopings. So again, this kind of loop anchor point or topological anchor point is kind of more or less same concept of the TAD boundaries. So we're using the high-C data to identify the topological anchor point RNAs. So we call it as a top RNA in short. So how they look like? So we checked pcRNA's localization in the TAD boundaries and also loop anchor point. So here, this is thick black bar here. This is the both boundaries, and here again this black bar is clearly indicate the loop anchor point. And when we do, this is not very clear, but anyway, the pcRNA is in orange or red, and its control linker, general linker RNA is in yellow. So as you can see, pcRNA have clear enrichment in the TAD boundaries and in also loop anchor point. So we think pcRNAs are more enriched in this kind of very important functional regions. So we identify these top RNAs, and this is one of the example. This is more familiar that UCSC genome browser fashion. So here, top one is TAD, the green. So this clearly shows where are the TAD and where are the TAD boundaries. And the second one is the loops. So the thin line is here, but each end shows very thick kind of bars where is the loop anchor point. And here is the very important HOXD gene clusters there, and this orange are our identified top RNAs. And when you zoom in, this is a high-C heat map, and when you zoom in, one of our examples is here, which is TOG. That's this promoter is inside of this loop anchor point, and it also have CTCF anchor chipset binding and zinc fingers binding. So this is, I will come back to this. So after we identify this interesting top RNA, we wanted to also characterize what are they. So firstly, even though we know that there is no maybe sequential conjugation, but we using slightly different approach. We use the sliding windows approach with local alignment algorithms, which is good to identify small patches. So indeed, we found this is the example of HNF6. This is a human, this is a mouse, and they do have same number of exons. And as you can see, even though not all of the transcribed regions is not very well aligned, but some patches are really well aligned. So we call this as conserved patches. So this was one of the examples, but I wanted to know whether this is also general phenomenon across the all top RNAs. So here is the x-axis is the scaled gene body. Top RNAs transcribed regions, because each one has a different length. And then this y-axis are all the top RNAs. And indeed, there are a number of groups. The first group, the blue ones, are some top RNAs that they have very good kind of conserved patches across the transcribed region, but some of them having enrichment in 5' or 3', or some have very little conjugation, but some doesn't have anything. So there is more or less 4 groups, and the important thing is that the well-conserved ones are more kind of developmental or functional related ones, and the one doesn't have anything is more or less kind of repressed functions. So another important thing I wanted to say is that the conserved patches are also important, but actually what are inside of these conserved patches? So we wanted to analyze motif search, and we found some enriched motifs. So this is the looping, and again, to remind you, this is CTCF, and there is high chance that on the other end of this loop, there is enhanced regions, and our top RNAs may be located like this. And then, firstly, we checked the motifs in conserved patches inside of the top RNAs, and we identified 32 highly enriched AIMer motifs, and we phylogenetically clustered them into 10 groups. And then we went to the TomTom database to check whether they are already known TF. And indeed, most of them are very well known. And then one striking result is that 8 out of 10 TF groups are the zinc finger protein groups. Zinc finger is very interesting one, because CTCF itself is a zinc finger, and zinc finger is already known that it can bind to the DNA and also RNA at the same time because it's dimer. So anyway, we also checked the other end of the loop, encode region, enhanced regions, and then we do see 3 groups are very enriched again, and these 3 are all again zinc fingers. So this is one of our interesting findings. And lastly, everything is look good, but what's that mean? What's the implication in human health? So we use the cancer data set because it's a well-annotated one and very nice data sets. So we used TCG RNA-seq data to do the pan-cancer analysis. There are about 34 cancer types, but we wanted to use normal samples as control, because if you just see the expression level in tumor, it's not really meaning anything. So unfortunately, 10 of them doesn't have any normals. So we use 24 cancer projects that have the normal samples. So it's reduced to 8,850 samples. So we did this comparison between the tumor and the normal samples, and when we draw the heat map, it turns out like this. So this, I just input the expression values, and I did unsupervised clustering, and it's classify all the 24 cancer types very nicely. Up here, this is all the TCGA cancer code. And also, some of the top RNA groups are really spread out. For example, some of the TAP RNA groups are really downregulated in glioblastoma, but some of them are highly upregulated in neuroendocrine cancers. So this is quite interesting results, and we do know the list, and we also published all the kind of gene names and link RNA, pcRNA names in our supplementary. And also lastly, also I did kind of cancer somatic mutation analysis. As you know, somatic mutations are everywhere, so we want to using very stringent condition, which is if this is CTCF, they must have a peak, and also underneath that there is a motif, and of course there is a TAP RNA. And usually the core motif is 19 to 20 moles, so the somatic mutation has to be inside of this small motif. So CTCF motif indeed have a lot of enrichment, which means CTCF motif in the TAP RNA have more chance to have cancer mutation, and also our novel motif, novel finding is zinc finger 263 has even better result in this case. So in conclusion, so in 3D genome, our identified TAP RNAs are located nearby the genes, and somehow this one regulate the genes throughout the zinc finger motif, particularly in developmental genes and also in cancers. So this work has done in Tony Kuja's group in the Cambridge University in collaboration with EBI. And thanks to everybody. And just for 30 seconds, I just completed my postdoc, and I moved to another institute in Cambridge, which is a Miller Institute. This one is all about the drug discovery research. And I'm now setting up a computational biology group in the institute. So the reason I'm talking about is I have 2 open positions for postdoc and a few PhD student positions. So I'm very happy to talking about it as well. Thank you. Yeah, so that's, I think, a really good question. So actually we did a number of other kind of analysis, but we actually didn't put in the paper. One of them is this isoform or alternative splicing analysis. That to be honest, I haven't seen John Mattick's paper because recently I moved my focus. But I think, we do have a similar kind of what is it, phenomenon or trend with,
So I have 2 questions about the relevance in cancer. So in your clustering, so did you try to take a random set of a similar number of non-coding RNAs or genes for the same purpose and redo the clustering? Because probably you will find that any set of genes will probably separate cancer types in a similar way.
We done kind of number of controls, but we haven't really see that kind of very nice clustering. We didn't see it, and also somatic mutation as well. Yeah.
And for the somatic mutation, so did you try to select a random set of enhancers, well-annotated enhancers, and see whether they have enrichment in somatic mutations? Because probably the enrichment that you're seeing is just basically related to enhancers rather than to these non-coding RNAs.
So sorry, I don't get the second one. So
no, but I'm wondering about your control. So you're comparing with somatic mutations in other non-coding RNAs, but did you select a set of enhancers, random set of enhancers, different from the ones that you are selecting with CTCF and the TAP RNAs? and see whether they also have an enrichment of mutations or not.
Yeah.
There may be, what you're saying is an association to enhancers rather than, or these enhancer elements rather than to the top RNAs.
Yes, yeah, I understand. Yeah. So actually, first part, when we identify pcRNAs in the pipeline, we also have number of steps to reduce the number down, and we get the confidence set. So in the middle process, we have a data set, a link RNA set that could be used as a control because they do have same length and they do have similar kind of exon structure. So we use this as a kind of control set. And we did the same analysis, finding CTCF motif inside this control link RNA and check how many somatic mutation happens. And that's what I showed in the bar graph, that we do have enrichment. I don't know whether
for the hierarchical clustering, I think that if you take any random set of genes, you will probably find a similar separation of the cancer types.
Okay, yeah,
We've seen that
okay.
before.
All right, okay, thank you.
Okay.